It's my honor to really be with the Foundation Fighting Blindness. I've been in medical research over 40 years, and this is truly an organization and a time in vision research that is moving forward at an accelerated pace. I think it's really important for everyone to know, as you have seen at this meeting, but we have a video of a lot of our researchers talking about translational science, because that's what we're going to be talking about this morning. And we think it's important that you hear from the people on the front lines, the people doing the work and translating all of the science that happens every day, 7 days a week, 365 days a year, into the preventions, treatments, and cures. So with that, if we can cue the video.
From bench to bedside, bringing treatments to patients.
For more than 4 decades, the Foundation Fighting Blindness has driven the basic research that has both furthered the understanding of retinal diseases and proven principles behind a number of potential treatments. Now is the time to take this knowledge and promise into the clinic, where treatments can be proven safe and effective and made available to anyone with a retinal disease. But the path to the clinic isn't without pitfalls. Here, in their own words, researchers on the front lines explain the challenge of translational research. To begin, the Foundation's Dr. Steve Rose tells us how the Foundation Fighting Blindness is uniquely positioning the field for success.
Translational research is, in fact, taking those basic discoveries and really learning how to apply them to these retinal degenerations. It's taking those discoveries, understanding what they really mean and how they can be brought to the individuals with these inherited retinal degenerations. It's the science behind understanding how you get it to the cells you need to get it to in the eye, and then getting it into a clinical trial so you prove that it's safe. That translation is what we call bench to bedside. It's translating the bench research, whether it be in a petri dish, in an animal, or whatever, into being able to deliver it to a human being.
But that's a really challenging process, and I think we forget sometimes how difficult that is. And of course, driving all of that is a huge challenge of raising enough money to support the research that's needed.
Researchers who work with the Foundation Fighting Blindness understand that it's not just a funding source, but also a partner in identifying promising opportunities and driving that research forward. Acting as a venture philanthropist, the Foundation often provides young, promising companies with what could be considered seed money, as well as a level of credibility, which they can then leverage for additional commercial support. The Foundation's Win-Gund Translational Research Acceleration Program has proven to be quite effective in this area.
We originally got a very small grant to work on a focused area of one of the inherited retinal diseases. When we got the Win-Gund Translational Award, it was $1.5 million over 3 years. But that's actually not sufficient to take a product from the bench, from the working in animals, to actually doing a clinical trial. However, it did function to leverage our ability to get venture capital funding, and we recently, just within the last couple of weeks, closed on a $37.5 million venture capital investment, which is sufficient funding to bring us for the next 3.5 years to work on 2 inherited retinal diseases plus another disease for a respiratory indication.
I really like to tell a story of how the research actually started that I'm doing right now, or that we're doing right now in the lab. And my collaborator and I, Craig Beeson and I, have submitted that same grant 3 times in a row to NIH, and it got turned down 3 times. And just about that time, Steve Rose from the Foundation had talked to me about how to best screen drugs for photoreceptor cell survival. So I showed him my grant, and he liked it. And 6 months later, he called me back and says, "We have money. Would you like to submit a grant?" And here we are 5 years later with a patent, a company started, and a really promising compound. And the foundation was willing to give us a chance and was proven right.
To me, that's where the foundation has been really great, is that they've sort of had the vision to say, let's fund basic research that we think is someday going to lead somewhere. We don't know exactly, we can't envision exactly how this is going to work out, but we have confidence, we have hope, and we have faith that these guys will get something useful out of this, and that other people can benefit from that.
We believe that it's important to bring these discoveries that the Foundation has funded for over 41 years through that translational research and get it commercialized so that any individual can in fact have access to that particular treatment or cure, but also then the foundation gets a return on its venture philanthropic investment in order to keep the science going, because we know this is not just a one-time shot.
And they've always held that as far as I've known the foundation and the people that are involved with the foundation, they've always had this belief, that they've had this confidence in the people that they fund, that their work will lead ahead to cures for blindness. And it takes that long-term kind of vision.
That vision has proven successful in one very notable instance: the first gene therapy clinical trials for retinal disease, through which more than 40 patients who were blind, including a young boy named Corey, regained some vision. For Corey, the results were life-changing. The Foundation Fighting Blindness not only funded the clinical trial, but every stage of research leading up to it. It is this success researchers everywhere are working to replicate.
That to me was groundbreaking. To be able to, I mean, first of all, it was the first successful clinical trial overall. And it was incredible to see the patients really improve significantly.
And in fact, it was Jean Bennett's Philadelphia-based team which actually realized that first clinical breakthrough. We asked her if she could recall how she felt when she first witnessed Corey's results and realized some sight had been restored.
I can't tell you what an amazing moment that was. In fact, it still sends goosebumps down my body thinking about it.
When I saw the interview of Steve Rose with the young boy who underwent gene therapy, and seeing him drive through New York and on the bus and be able to see what's around him and see his face afterwards smiling, that was just incredible.
But since then, I've heard so many stories from all of the different individuals, and the effect is the same each time. You know, whether it's from seeing fireflies for the first time to being able to see a mother's face clearly for the first time, to be able to see a long-lost relative and see them smiling at you from across the room, to being able to sit in a classroom and read a book. These are just miraculous things, and I feel so lucky, and my teammates feel so lucky to be able to witness them. We want this to be made available to anybody who could benefit from it, and we're very optimistic that we will be able to get this through. In fact, it may be one of the first gene therapies to be approved, and we're hoping that it could lead the way to many other gene therapies for other blinding diseases.
There's already 50 to 100 people in this country who have benefited from gene therapy. There are going to be hundreds of people in clinical trials. There is not just hope in the indefinite science fiction future. There's hope today, because real progress is being made in real living human beings to bring treatments to the clinic.
And our ability to move that forward is extremely important. And it's what drives me day and night. It's why I work the way I do. It's why the foundation works the way it does, because we know we need to do it, and it is an exciting time. We're seeing advances in ways that weren't seen even 5 years ago.
We're at a very special time, I think, in the history of medicine, because this is not just retinal diseases we're talking about. We're talking about many, well, it's basically all forms of inherited human disease are potentially amenable to this kind of approach.
I tell the folks in my laboratory and all the time, and people that I train, if I can just be a part of the process that one day gets a patient who is fighting
a disease like retinitis pigmentosa, to see better or to preserve the vision that they have remaining, then what more could you ask for in your whole career?
We are making a difference such that a parent, an affected individual, a child, should never have to hear you will go blind. All they will hear is, you have a retinal degeneration, we've got a treatment for that that will make sure you don't go blind. We just need the resources to be able to do it.
So you've heard from researchers throughout the world talking about how the foundation has made a difference. I'm extremely honored and pleased to introduce Dr. Phil Sanderson from the National Center for Advancing Translational Sciences at the NIH. He is in charge of the therapies for rare and neglected diseases. And just a quick aside, the head of the National Center for Advancing Translational Sciences, Dr. Chris Austin, actually trained in retina with Connie Sepko at Harvard. So he knows the retina field, he loves the retina field. Sorry he couldn't be here today. He really wanted to come to be able to address you, but we are certainly not losing a step by having Dr. Sanderson join us from NCATS and the TREND program. He also spent many years at Merck and really understands what it takes to bring treatments to individuals. With that, Dr. Sanderson.
Well, thank you, Stephen. You're way too kind. And this is really an honor to be able to come here and represent the NIH and NCATS and talk to this audience, which is a strikingly well-informed audience just from the evidence of having breakfast here this morning. So just a word about the NCATS, which is the National Center for Advancing Translational Sciences. What is it? Well, it's been around for about 18 months. It's one of the centers of the National Institutes of Health, and it was formed to help provide an answer to a very big problem. It's actually a series of very big problems. But I just want to say, we use to talk about the word translational. In a sense, translational, it's almost as if it's a new word or it's a new concept, and actually translation of basic research into something that is useful in people has been around for as long as people have been doing scientific drug discovery. It's just raised its head, the word and its use now, because there has been a change in the way that people are organized and funding and going about doing translational research. There's been a sort of a withdrawal to some extent by major pharma and the biotech industry from that early stage of translation. And so consequently, organizations like NCATS and other ones are being formed to fill and help fill that vacuum. So there is, and talk about this big problem. And in one sense, it's a very good problem. And that is that over recent years, and you've heard all about that in the previous sessions at this meeting and at previous meetings, tremendous advances in science, say, for example, the genetics, the Human Genome Project, advances in stem cell science. And over let's say the past 20 years in my career, there have been very, very remarkable advances in medical development. For example, I was the development of treatments for HIV is something which on many levels is truly remarkable and has actually revolutionized that disease. And you could say the same about other diseases as well. But the model for how those sorts of advances was possible is changing. And the problem is that the way that those sorts of advances have taken place are basically inefficient, relatively slow, and as a consequence, expensive. And part of this is due to the realization that all disease is a rare disease. Virtually, I'd say virtually all disease is a rare disease. And that has complicated the drug discovery and development arena. So at NCATS, it was the concept of NCATS was that it would complement the work of others, and that it would catalyze the generation of innovative methods and technologies, this is from our mission statement, that will enhance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human diseases and conditions. The whole concept being to find a way to work with people to do drug development faster, better, and cheaper. And along the way, NCATS is interested in discovering and developing new modalities, new treatments, demonstrating that they work, and then disseminating our findings so that it can be taken, so that our learnings can be used by others. So we collaborate, it's highly collaborative, it's not a competitive business. We will collaborate with academia, advocacy groups and foundations like the FFB, nonprofits. We have good contacts with the FDA, and we collaborate with pharma and biotech as well. We'll collaborate with just about anybody. Now, it's a big, complicated project. It has many different components and facets to it, and I won't be able to talk about all of them today. I just want to give two big programs that are taking place, organized, at least in part, through NCATS. And these fall under the kind of rubric of re-engineering translational sciences, really making a very big fundamental change in the way that translational science is taking place. And the one, I'm going to highlight two. One is called Tissue on a Chip, and that uses the new advances in, particularly in stem cell, essentially cell technologies, to be able to replicate human tissue, to build human tissue on a chip. And this has two functions. You could use that to test on a chip for efficacy, basically function and efficacy, and you can also use it as a measure of safety. So this would, if successful, completely change the way that preclinical efficacy and safety research would take place. This is a big project with a big goal at the end of it. This is done in collaboration with DARPA, the Defense Advanced Research Projects Agency, collaboration with the FDA, and with organizations such as the Wyss Institute at Harvard. And we have given out grants now, I think it's 17 grants for work focused on developing tissues on a chip. And the second one is, and again, we've the organization has just given out its first grants for this, which is discovering new therapeutic uses for existing molecules. This is very different, but it's getting the pharmaceutical industry to do something which they've never done before, which is to give up their molecules that they've tried to develop, that have gone into the clinic and have been demonstrated to be safe in people, but did not work for their given disease, or were discontinued simply because, simply for business reasons. What they have done is that they have decided to pool and share these molecules. There's 58 of them, and it's 8 pharmaceutical companies, so that academics, sort of a crowdsourcing idea, can apply to NCATS to work with those molecules to see if they can be used for other diseases. And there are so many rare diseases out there. If you come to NCATS with a good idea and it passes a review, and a very rigorous review, then NCATS may fund those sorts of studies. So with that, I'm just going to narrow down now from those big picture programs to something which is closer to what FFB is interested in. And part of what we do can be described as being re-engineering translational sciences, and that's a whole myriad of different problems. And there's many different ways in which you can help to translate basic research into the clinic. At the early stage there, it's like the gateway into drug discovery and development is something which takes place at what we call the NCGC, the NIH Chemical Genomics Center down in Rockville. They have currently greater than 200 collaborations across the world involving such technologies and techniques as assay development, high throughput screening, chemical informatics, essentially computational techniques, and medicinal chemistry, trying to go from what you might call a target. It's a biological mechanism which has a potential, somebody believes has a potential, to turn it into something which has a degree of proof of concept in the test tube. And the focus tends to be on unprecedented targets, very difficult ones, things that other people have not had success with. Try to use new technologies, all the best biology and technology out there to try to turn the idea into a reality. And there's also a focus on rare and neglected diseases. So I'm going to talk about two programs eye-related within that group. And the first one is a collaboration between a fellow called Jim Inglaze at NCATS and Don Zacks, that's a name I think some of you might recognize. And this is the identification of a new drug target for the prevention of glaucoma-associated vision loss. And the goal of this is to find a drug lead which promotes survival of retinal ganglion cells, the RGCs. They're the cells on if you like, the inside of the retina that actually communicate directly into the brain. To find a drug which promotes the survival of those cells and prevents vision loss caused by glaucoma. And at Johns Hopkins University in Don Zacks' lab, they identified a drug class. They're called kinase inhibitors. It's a widely recognized drug class within drug discovery. They identified a drug class which looked like it had promise for promoting retinal ganglion cell survival. They then turned to NCATS for help in two areas. And the first was to identify specifically the target. They had a kinase inhibitor which seemed to work, but they didn't know why. To determine what the target is, they turned to a collaboration with a fellow called Scott Martin, who's an expert at short interfering RNA technology. This is it's a sort of a genetic technology, if you like, where you can specifically shut down the action of any gene to see if that would reproduce the effect that the kinase inhibitor produced. And they were able, and he screened all 623 kinase genes, the products of each of those kinase genes, and was able to determine that it looked like there was a specific kinase called DLK, dual leucine zipper kinase, which was the kinase responsible for the cell death. If you inhibit that kinase, you can prevent cell death. What NCATS also did, and this was in a collaboration with Jim Inglaze, was to then screen a whole battery of known kinase inhibitors to optimize the early stage of the drug development, to find the optimum tool compound. And they did that, and they were then able to take one of those kinase inhibitors, which inhibits DLK, take it into a rodent model of glaucoma, and they showed that it actually protected the RGCs. So there you have an example of early stage. You take an idea, you using the translational expertise, the early translational expertise at NCATS, you can identify and validate a target which could then be picked up for further development, further refinement and development later on. A second project that I'll talk about has to do with stem cells, and this is a project between Mark Ferrer and actually Kapil Bharti at the National Eye Institute, and this involves stem cells promoting induced pluripotent stem cell differentiation. It's promoting the differentiation so that to look for a molecule which increases the efficiency of the differentiation of the induced pluripotent stem cell into mature retinal pigment epithelium, or RPE cells. Now, if you take RPE cells derived from induced pluripotent stem cells using known methods, they look and function like mature RPE cells. But they have a pattern of gene expression. If you kind of look inside, they have a pattern of gene expression which has some stem cell character. And that raises the question of whether there might be a risk. And it's unknown risk, but there may be a risk with that stem cell character that you could have teratomas, essentially unwanted growths, in the body if you used such RPE cells generated by known methods. And so what Mark Ferrer in his lab is doing is developing an assay and a screen so that you can find small molecules which in the test tube will reduce that stem cell character and will actually turn an induced pluripotent stem cell into what it is in effect is a mature RPE cell without that risk associated with stem cell character. And that work is underway. So, now moving on a little bit later in the development process, in the preclinical development process, I'll talk about the Therapeutics for Rare and Neglected Diseases program, which is something that I'm part of. This is a model where it's a comprehensive drug development collaboration. So we put out solicitation, we solicit ideas and projects. You can apply to Trend, which typically it's a solicitation once a year. And if a project it's reviewed, a very, very rigorous review process takes place. The project can be then taken accepted into NCATS. We're not a grant-making body. We actually are a group of scientists like myself who have expertise in drug discovery and development. And we will then use NCATS resources and contract resources to advance a project. And it's typically going to be a project where you've got some kind of proof of concept in an animal, a little bit like the Don Zacks program. And you're now going to take it all the way to IND, new drug application, with the FDA or some equivalent body outside. And we may even support it in the clinic, up to, at most, phase 2A. We'll take small molecules, protein, cell therapies, and what have you. The disease must meet rare disease criteria, or actually WHO neglected tropical disease criteria. And the program serves to develop and demonstrate sort of new ways, new paradigms, and more generally applicable platform technologies as well. We'd like to be able to help specific projects, but we'd also like to be able to take on projects where you can use your results to help other researchers who are in related areas as well, have a general applicability. There have been 17 projects adopted since 2009 into Trend. 4 drugs have been taken into the clinic right now. To give you an idea, there's a leukemia drug, something, the prototypical genetic therapy or genetic disease is sickle cell disease. Have a program in the clinic for that. And then a myopathy, and then Niemann-Pick C disease as well. And we also do natural history studies, and I'll come back to the importance of natural history studies in a little bit later. There's a second program called Bridges, which I won't go into. It's a little bit narrower in scope, but it's been around a little bit longer. Its funding mechanism is a little different, but there's been 20 completed projects of that. 13 out of 13 projects for which there was a submitted IND have actually been cleared. 12 agents have gone into phase 1 clinical studies, and 3 so far have made it to phase 2. So, to give you 2 specific projects that have just been taken in, just been accepted into trend. They're not even up on the website now, so you're the first public people who are aware that these have actually been taken in, and I almost ran into trouble when I said that I was going to be talking about this. But we have a collaboration with a company called Pharmaceuticals to develop what's called a small molecule, so that's a regular drug, you know, small drug like aspirin, Pharmacological Chaperone for the treatment of class 2 autosomal dominant retinitis pigmentosa. Okay, so let me explain what that is. So, well, you don't need to know what retinitis pigmentosa is, but pharmacological chaperone is something which you'll need some explaining. First of all, class 2 autosomal dominant retinitis pigmentosa is characterized, it's autosomal dominant, meaning that it's carried on an autosome, it's not sex-linked, and it's dominant. In other words, there's a gain of function which overrides the normally functioning gene. Class 2 autosomal dominant retinitis pigmentosa is characterized by mutant opsin. So it's the opsin gene, which is mutant. It produces a defective opsin, which is prone to misfolding, and that leads to 2 things. The first is that it doesn't go where it needs to go, to the rod outer segment, to then combine with retinal and give you your functioning and working rhodopsin. But the second thing, which is actually more serious, is that it aggregates in rod cells, and it causes, those aggregates it just accumulates and causes toxicity, and ultimately leading to cell death. The cell death then triggers, it's a sort of a cascade a slow motion cascade of processes leading to retinal degeneration. So the symptoms, as you know, is loss of night vision and side vision at minimum. Roughly about 15,000 patients, it's believed, carry these types of mutations, it's more than one, around the world. So a pharmacological chaperone is a molecule which can correct that defect. Now, it doesn't correct the gene. What it does is, by a mechanism that's not truly unknown, but it's truly known, is that it will bind to the folding gene, to the folding protein, and to promote its folding into the correct shape. So it is then recognized by the cell's transport machinery to go to the right place, to the outer segment. It can bind retinal and then function as normal. Now, the Pharmaceuticals have designed a small molecule which will do that reversibly. It goes on, promotes the folding, gets it to where it needs to go, and then comes off. It has moderate affinity. If it was too high affinity, it wouldn't come off. And it has relatively short half-life. So again, that's important. It goes away so that then the molecule comes, the chaperone comes off, retinal can bind, and you form the pigment. Now, this lead molecule has been extensively characterized by BICAM preclinically in models. It promotes rhodopsin formation. You can show that in cell culture. It gets to the right place. More than that, in mice which contain that mutant gene, it actually preserves rod cell viability and function in mice after oral administration. So the potential is there for an orally administered drug which could be used to treat class 2 autosomal dominant RP. So they applied to Trend, and we are here to take a look at their program. We've taken it in. We're taking a very careful look at their program. Repeat some of the studies that need to be repeated to make sure that everything is good. And what we want to do is to develop a lead molecule and then also identify a backup molecule in case something bad should happen to the lead molecule. And we also would like to initiate a natural history study of class 2 autosomal dominant retinitis pigmentosa. Now, you know that retinitis pigmentosa is a slow motion disease typically. It takes many years for it to develop. This has a half-life, if I understand it right, of 7 years in terms of the degradation of the ERG. If you're in a drug development program, you don't want to have to wait that long to know whether your drug is working or not. And so what we want to do, and we're talking with the NEI and others, is to find a way to use the latest technologies to measure the decline of the function of the retina so that hopefully when we get into the clinic with this treatment, you can tell relatively quickly whether it's working or not, whether then somebody would then be much more willing to put up a lot more money to expand the clinical trial and accelerate its movement towards approval. So that natural history study may be an important component of accelerating and speeding the drug development. So one final project before I finish up is a collaboration we have with the University of California at Irvine. And this is a collaboration involving using retinal progenitor cells for the treatment of retinitis pigmentosa. These retinal progenitor cells are sort of part differentiated stem cells. They're on their way to being the final end product, if you like. And they can differentiate. They won't differentiate into cells typical of the rest of the body, but they can still differentiate into cells which make up the components of the retina. So the candidate cell line that our collaborator has come to us with has shown feasibility of transplantation in animals. The cells survive in the vitreous. This is after injection, and they do migrate to the retina. Efficacy has been demonstrated in a rat model. This is the standard rat model which is used. It's the RCS rat. And they've shown rod and cone preservation and function. And safety studies have been initiated. And actually, that's an understatement. There's actually been 9 months of safety demonstrated so far, and with no concerns raised. So they've come to us. We've just started talking with the collaborator. We're performing, again, a gap analysis. What still needs to be done to complete it preclinically to move this forward? And we're going to put together a project plan in collaboration with the collaborator. And we want to be able to support them to be able to complete the studies needed for filing IND. And we also want to take a longer view also with this technology because stem cell technologies is so early and it's so risky going into phase 1 that there's many different things that could go wrong, and we're taking the view that there are some preclinical studies that we can do to assist with the long-term prospects of the technology. And so with that, I'm just going to say that is, I hope I've given you a flavor of 4 different projects that we have, too early, to at the stage where we can really think about getting the compounds or treatments into the clinic. NCATS has a website which you can, NCATS, just Google it and go from there. And at the end, I'll be happy to answer questions.
Thank you, Phil. very, very enlightening, and you heard, and I'll actually talk a little bit about a couple of those projects. The Foundation Fighting Blindness started in 1971, 42 years ago, when, as we heard numerous times throughout this meeting, there really wasn't anything known about retinal disease. But even at that point, the foundation took the long view that Phil was talking about and understood that in order to get treatments to those individuals who are affected by retinal diseases, you had to take chances, you had to move forward, you had to do what wasn't called in those days translation, but actually was. And how did that start at the foundation? Well, you actually heard the culmination of that in the Argus II presentation. The foundation in its early days supported Mark Humayun and Jean Dewan at Johns Hopkins University when the idea for a retinal prosthesis was just in their mind's eye. And that shows that even back then, at the beginning, the foundation understood what translational science was. As you heard in the video from Jeff Choulet from Applied Genetic Technologies, the foundation also understands the importance of leverage, something, I'll be honest, I didn't understand till I came to the foundation. I'm just an academic. The business people on the foundation have taught me about leverage. And that's where the foundation's support for translational work, in fact, can attract outside investments. And as you heard from Jeff, our support of AGTC brought in $37 million for translation in gene therapy for achromatopsia and X-linked retinoschisis. Very important for the foundation in order to maximize the investments that we make with your dollars in order to get treatments to the clinic. It is that long-term vision that has moved the foundation forward to the point that you're now hearing about treatments that the foundation have been involved in the basic science for many years. A few examples: Usher 1B, the gene therapy trial from Oxford Biomedica called USHSTAT. The foundation funded the basic work done by David Williams at UC San Diego and UCLA and others in order to allow Oxford Biomedica to bring that treatment to the clinic in an ongoing clinical trial. In the same way, the foundation funded the basic research to bring StarGen, the Stargardt clinical trial, also by Oxford Biomedica, in order to get it into clinical trials. So that's where the foundation has always understood the long view, the understanding of what it's going to take to get treatments into the clinic and translate them into possible cures, preventions, and treatments for inherited retinal diseases. We heard Phil talk about Don Zach's work within the NCATs. Don is a well-known researcher to all of us at the foundation. You've heard Don talk before. Phil talked about Don's work in retinal ganglion cells, and some of you are out there scratching your heads going, why do I care about glaucoma? Well, it turns out the system that Don is using to analyze his cell-saving potential treatments has direct applicability to photoreceptors. And in fact, that's what the WinGun TRAP program is supporting, is in collaboration with NCATs. Don is moving forward in investigating these molecules, not only for glaucoma, but also for retinitis pigmentosa, choroideremia, Stargardt, and other inherited retinal degenerations. You heard Phil talk about the long time frame it takes to do clinical work, preclinical research, translational research, and certainly clinical trials. I just had a discussion with Randy Wheelock from the Choroideremia Research Foundation about the frustration that I know all of you feel. As Howard Hirsch, one of our board members, once said, it seems like research happens in dog years. Well, this is where, under the guidance of Dr. Patricia Ziliaks of our Clinical Research Institute, we're investigating new clinical endpoints, a different type of translational research, a type of translational research that will shorten and make less costly clinical trials. You heard Phil talk about the half-life of retinal degeneration being 7 years. The important point, as Phil pointed out, and certainly from Patricia's history in industry as well as Phil's, we all recognize that pharma, biotech can't afford 7-year clinical trials. It is the translation of our knowledge through Patricia's guidance that we believe is going to lead to a much shorter half-life, of much shorter ability to determine when, in fact, a treatment has had a positive effect, and therefore draw pharma into the areas of inherited retinal degenerations. As we all know, pharma has withdrawn from research and development, early stage. That's where the foundation's support, as talked about by Jeff with leverage, and as we're doing as the foundation, moving to partner with pharma in order for our research to be able to provide that necessary proof of principle work that's going to interest pharma to take these rare inherited retinal degenerations forward into the clinic. It's an exciting time. It's an important time, and partnerships between the foundation, groups like the National Center for Advancing Translational Science, as well as pharma and biotech, are really going to bring these to fruition, and you're going to see that happening in an ever-accelerating pace through the foundation's efforts and your support. I want to thank all of my colleagues. I want to thank Patricia for all of her work. Without it, we wouldn't be moving as fast as we are. My crew, Amy Laster, Ben Shaberman, while the blog has my name on it, Ben is my writer. So my hat's off to Ben for making things possible for people to understand and to keep you up to date. And with that, we will take questions. I have a question in back. Eddie?
Regarding the NCATs, are we limited to just US-based companies being involved, or is it global from a standpoint of getting companies involved?
Are the companies involved with which?
As far as the things that you review and try to put people together.
Oh, it's global, anybody.
Okay.
Yep. There's a subtlety in one of the programs that I talked about, which is Bridges. If it is outside of the U.S., it has to be something which is a small, what would classify as being small company by U.S. standards. Trend will work with anybody.
We have one over here.
Microphone's coming to you.
Yes, I was curious to know what the impacts of sequestration and all the budget talks are on what's happening in these federal research places.
That's interesting. Overall, I'll just say for NCATs, this is something which I'm reasonably familiar with. I don't deal with the overall budget, but overall, the money that the funds that come from the government to NCATS, the vast majority of it goes out in terms of contracts and grants from areas of NCATS which I haven't been able to talk to you about. The impact of the sequestration is that the recipients of those are the ones who suffer the most. So now, as it happens, so if there's a 5 or 6% cut, that means that there's 5 or 6% less money to go out in grants, 5 or 6% less money that can go out in contracts. I would say that NCATS was very forward-looking over the period when it looked like the sequestration was coming, and they did, from what I can tell, an excellent job in preventing, essentially, planning for the worst.
The one other comment I'd add, unfortunately, is that it has led to a $40 million decrease in the National Eye Institute budget, which has had an impact, in fact, on grant funding to institutions. Depending upon what part of the program you're in, it can be anywhere from 5 to 7%. And while that doesn't sound like a lot, it actually is to a researcher in keeping the work going. We have a question over here.
Yeah. With regard to NCATS, when you find something that was a rare disease and you believe you have a treatment, if it doesn't quite work on the rare disease, but it does work on something larger like macular degeneration, can that still go forward?
Well, so we would do everything we can to see that it goes forward. If it does not meet the rare disease criterion, then we will do everything we can to point the researcher organization in the direction of somebody who would be able to do that. Yes, I mean, that's part of our job, is seeing that these things move forward. And being part of, being part of the NIH means that we're very well positioned to find, partners for that type of situation.
Other questions?
Dr. Rose, I have a question, or ask if you could describe the relationship between FFB and CATS, maybe with some examples, just maybe like a day in the life of, or a conversation. How does that work?
Sure. Probably the most tangible example is Dr. Don Zach's work. So Don came to FFB with an idea based on Don is a glaucoma clinician, okay? So his work is really based in understanding glaucoma and trying to treat glaucoma. It turns out we hijacked Don and got him interested in inherited retinal degenerations. And during conversations with Don, it became clear that there was a possibility that the work he was doing to try to preserve the cells that Phil was talking about, the ganglion cells, which are part of the retina, they are the cells that transmit the signal from your retina to your optic nerve, which goes back to your brain, that in fact molecules that might keep those cells alive in glaucoma could also be applied to inherited retinal degenerations. So we discussed how we could use his work in glaucoma to investigate what would happen in photoreceptors, other retinal cells, as well as his glaucoma work. That's where I used to work at NIH. I went down and I had a discussion with Chris Austin, with Don Zach, and we said, okay, how can we work together? The NCATS has a huge industrial strength screening program for molecules that's robotics. If you go to the NCAT site, I believe there may even be a video there that shows these robots taking plates and moving them and doing all the assays. And our thought was, how can we, again, from my business lessons, leverage what the NCATS has in infrastructure for our purposes? So we are actually funding Don, who has a postdoc who is working with NCATS in order to screen molecules using the glaucoma system, because that has a better readout for an automated assay. But then he brings his positives back and then screens them in a less automated way for the inherited retinal degenerations. So that's how we have made a marriage between FFB effort and the NCATS effort, using the infrastructure that NCATS has in order to advance our field. Other questions? Right here in front. Mike's coming to you.
Dr. Rose, thank you. Nikhil Parekh. I actually work for pharma industry, so first comment. I think the disappointment with a lot of public in pharma getting out of the translational type research is because of what I call efficiency of research. If it takes too long, nobody wants to invest it. So that's a comment. But given that comment, how does pharma industry know that there is a need like NCAT where old molecules have not been, can be studied into different models so that they can be tested for other conditions that not been tested for? And the second thing is, how does the NIH and say, for example, NSF or even CEDAR or CBER work all together, because pharma industry doesn't know what molecule FDA would allow to progress to different disease state?
Well, to answer your first, I'll take the first question. So, pharma has historically been very insular, in the sense of each company very insular. And there is a belief, and if it's still propagated by former senior pharma types, that if the new use wasn't found in their particular, in that company, then you'll never find one, because we do the best job. Now, that's one part of the component, one component. The other component is that they're afraid essentially they're very possessive of their molecules, the molecules' properties, and all the data that they have. Because there's a feeling that could be, you could be giving away the farm in some sense, or this data could be misused. And so there's a barrier, two barriers really, to overcome. So what this new initiative was able to do was, first of all, admit that crowdsourcing could be a good idea, and we'll see just where that takes. We'll see if it really truly is a good idea. Number one, and number two, that just get used to the idea of sharing. It sounds really, basic, but that's what it is. There's a bit of a risk there, but there's benefits too. Nobody knows how well that's going to benefit all of them, but they're in a position where they need to take that risk now. The third thing is that sharing involves there's a big business, legal, intellectual property issues that need to be worked out. And as soon as you start to collaborate with somebody, especially in industry, between companies, that is an enormous headache. Actually, much more so than for an organization like the government. We're much more open to collaborating, and it's much easier, although not trivial. So that's my take.
So the NIH director called in the CEOs of quite a number of big pharmas for meetings, and a number of them took him up on his proposal to put these molecules into the NCATs and allow that. It's called pre-competitive sharing. The case has been made that better to get something out to the public than to have it get cobwebs in a vault. And that the best place to do that was the NIH, which is a, if you will, non-denominational entity. Its job is for the public health. As far as people, pharma, understanding what's going on, that's actually been widely broadcasted by the NIH. It's on the NIH website. So again, as far as the foundation goes, we have told our investigators to apply to the program. We honestly, because of the way the constraints around government, we can't go to the government and say, well, who applied and who didn't get it. We only know who does get, and we make our investigators aware of what's out there. The list of molecules are available, their properties are available. It could be an investigator applied and didn't get for whatever reason, or it could be some, in looking at those molecules, made the decision it doesn't fit within their purview of their research. One more question if we have one, and then we've got to go. We have right here.
Hi, I was wondering why, if advanced cell technology already has the ability to derive RPE cells from embryonic stem cells, why are we also trying to derive RPE cells from skin cells, or however that's done? And is it because there's no sharing in that regard?
So there are a couple of answers to that. You hit on one of them, which is each of the companies that are working in this have a proprietary interest. But more importantly, scientifically, if you look at the cells that are made by the different groups, whether it be ACT or the London project under Pete Coffey in conjunction with Pfizer, or other groups that are doing it, the Riken Institute in Japan, Dennis Clegg at UCSB, and others. They're actually using slightly different methods of doing so. And one of the things that needs to be done is to really characterize those RPE-like, let's be clear, I use that term very specifically, RPE-like cells, and characterize them against standards of regular RPE cells to make sure that they all function the way they're supposed to. RPEs have polarity. There is an upside and a downside. Are we producing them in the right way such that the upside does what it's supposed to do and the downside does what it's supposed to do? The second part is, in the ACT trial, they injected a bolus of cells. And those cells are maintained as a little ball of RPEs right where they injected them. Well, we know the RPE is a layer on the retina. So the other groups that are working are looking to put in sheets of RPE, whether on a biodegradable matrix, on a non-biodegradable matrix, or a sheet without any matrix at all. This is still in the investigational stage. What is going to be the best way to deliver these cells so they perform their function? And that's what's going on. Is, as in all science, there are lots of different ideas. You've got to prove it. And with that, we will conclude the session. This is the last session for Divisions 23, but the registration desk will stay open until 1 p.m. in case you need any assistance. I want to remind everyone that you have an evaluation form in your conference bag. Please take a few minutes to fill those in. It's really important to us, as we move forward for Visions 2014 and beyond to understand what you want and what you get out of these conferences. So your feedback is very important. For those of you attending the National Trustee or Board of Directors meeting, those will be held in Grand Ballroom 7 through 10. I hope everybody enjoyed the conference experience, and please have a safe trip home. Thank you.